Hello, everyone. Good morning, good afternoon, good evening. If you weren't here yesterday, this is the second day of our microbiome data analysis course as part of our series. I'm Sean Gibbons, a faculty member and a microbiome researcher at ISB, or the Institute for Systems Biology. ISB is a nonprofit biomedical research institute located in Seattle, and we're an affiliate of the Providence Health System. We focus on some of the most pressing issues in human health, including COVID-19, brain health, aging, cancer, the microbiome, sepsis, and many other chronic and infectious diseases. ISB also focuses on improving K through 12 STEM education and beyond K through 12, as you can see, in particular by addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn more about ISB by visiting our website at isbscience.org, which you can see behind me after I get up and leave here. Okay, so before we get into it, I'd like to acknowledge that we are on the traditional unceded land of the coastal Salish people, including the Duwamish tribe, past and present. We honor with gratitude the land itself and the Duwamish tribe. In addition, I'd like to recognize our generous sponsors who have made all of this possible, in particular Illumina, Thorne Health Tech, and the Washington Research Foundation. And you'll be hearing from Illumina and Thorne Health Tech later on in the day during the lunch break. Because of their support, we've been able to provide this virtual course and symposium free of charge to at this point, over 2,200 people from about 80 countries and about 46 states. To all of you around the world who are joining us, we're grateful for your time today. We're excited for this opportunity to help train the next generation of microbiome researchers from around the world. If you are on social media, please remember to share your experiences about the 2021 ISB Microbiome series using the hashtag #ISB Microbiome 2121, all one word, on Twitter, Facebook, or what have you. And if you want to tag ISB as an institution, their handle is @ISBSCI or ISBSci. Okay. So at this point, yesterday we did a poll to kind of look at who was joining us from all around the planet geographically. Now today we want to ask you a little bit something about yourselves, about your background, who you are, whether you're a student or a postdoc or a PI. So we're going to put that poll up, and please participate and let us know who you are. And at the end, we'll kind of look at that, and I'll get back to you on what the demographics are of who's joining us here today. So to kind of give you a brief review of what we did in the first day of the course yesterday, Dr. Christian Diener, our brave instructor, walked you through amplicon sequencing data analysis using the QIIME2 ecosystem of software tools. And that course and all those materials still exist on our website. You can access all of them, the notebooks, the slides, and the video is also accessible, which we've posted to the Slack channel. And in the comment of the Zoom here, we're going to post links to the Slack and to all these other things as well. So you can get access to all of that. So you kind of got the basic tool set for interrogating these really, really common data sets that we tend to deal with in microbiome research, which in particular is the 16S gene, amplicon sequencing of this particular gene to look at who's there in a particular ecosystem. The second day of the course is going to be quite different. So if you remember last year, we did a little bit of metabolic modeling using this tool that our research institute has helped to develop called MyCom. Indeed, Christian Diener was the developer of this tool that allowed for the kind of simulation of the metabolism of entire microbial communities. This year, we're going to zoom down into building metabolic models of individual isolates of bugs isolated from a human gut. And we're going to leverage, I forgot to mention that yesterday in our course, we actually leveraged an open access data set from the Earth Microbiome Project. So this EMP data set is we took samples from this to analyze in that course. Today's course, we're also going to leverage an open resource, so open data resource, which are whole genome sequences from the BioML strain library. I believe it's the Broad Open Biome Microbiome Library, which is thousands of strains that have been isolated from the human gut and had whole genome sequencing done on those individual isolates. And so we have a list of many, many strains, and you as a student today are going to construct your own metabolic model from one of these strains. And then we're going to kind of compare across strains and look at differences between them based on these models. So the first part of the course is going to focus on model construction. And then we're going to get into a really interesting topic, which is reverse ecology, whereby you use information encoded in the genome to try to back out what the ecological niche of a given organism was or is in that environment. So Christian's going to explain this to you today, and that's going to be a lot of fun. And we have a special treat. At the end of the day, our very own Priyanka Baloni is going to give a research talk towards the end of the day that moves away a little bit from microbes and towards the human body, and kind of looking at how microbial metabolism affects metabolism of the human host, and how those interactions can potentiate disease or cause issues in the body. So kind of bringing a sort of system scale perspective on what you've learned here today. And then again, this will all kind of set us up for tomorrow, which is our symposium, where we have 6 speakers from across the field who are focusing on the evolution of commensal microbes and how genomic variation affects function of these strains in the human body and how that might impact health. So really excited about today and about tomorrow. Our team, we have the same great team of TAs that you had yesterday here today. Tomasz Wolmanski, Noah Rappaport, Alex Carr, Priyanka Baloni, and myself will be on the Slack channel there answering your questions, helping you as Christian lectures. We also have an incredible administrative support team. Our fearless leader, Joey Petosa, Allison Kudla, Joe Meister, Audrey Hubbard, Renee Duprell, Thea Swanson, and Adam Myers. So thanks so much to all of these folks who've come together to give their time and their expertise to make this event what it is. If you have any issues with course materials or need assistance during the course, please visit the Get Help Slack channel or the course Slack channel. You've already received this link in an email, but we've shared it again in the chat of the Zoom. To ensure everyone can enjoy this course in a harassment-free environment, we've also provided a code of conduct this year, which can be found in the general channel of the Slack. If we observe behaviors that violate this code of conduct, we will warn people, and we reserve the right to remove folks if they're bad actors. This didn't happen yesterday. Everyone was great and very positive, and I don't anticipate this happening today either, but just wanted to let you know that code of conduct is there. Okay, so I guess right now I will take this time to look at our demographic poll. So who is joining us here today? It looks like the winners, as far as like numbers of frequency of who's joining us, are graduate students. So about 43% of the folks who are Zooming in right now are graduate students. Next are postdocs. 24% of our participants today are postdocs. Then it looks like it's a close tie between principal investigators and undergraduates. So about 9% of the folks joining are principal investigators, 6% are undergraduate students. We see about 5% industry scientists, 3% educators, 2% clinicians, and then we have a catch-all. So 9% of folks are reporting that they are some other kind of microbiome curious person that's out there. So that just gives you an idea of who's all on the line today. Welcome to all of you. We're so grateful for you being here, and we're excited to have you with us for the rest of this day. It's going to be a great course. So without further ado, I'm going to end my introductory comments now. Soon you'll be hearing from Dr. Christian Diener. the very talented computational biologist who has designed these courses and will be teaching you today and taught you yesterday. So we'll have a few minutes now between me ending and him beginning. The course will officially start at 9:30 Seattle time. All right, welcome and thank you.